Thank you for the introduction, and good morning, everyone. So today I'm going to talk about a network propagation approach that we have applied to the study of rare genetic variants in sporadic Alzheimer's disease. So, yeah, genetic variation, as you know, takes many forms. For the purpose of this talk, we're going to focus on SNPs, meaning single nucleotide polymorphisms, which are basically changes in a single letter in the DNA sequence. And SNPs occur in the population at different frequencies. So based on the frequency, we talk about rare or common SNPs. And rare SNPs are the ones that occur in the population with a frequency of less than 1%. Also, in relation to complex traits or diseases, we know that there is an inverse relationship between SNP frequency and effect size, so that typically rare SNPs have huge effects on complex traits or diseases. And we also know that rare genetic variation has a big impact in the context of neurodegenerative disease. And just to name a few, we can mention the rare mutations in the APP and presenilin 1 and 2 genes that cause familial Alzheimer's disease, and also the mutations in the MAPT, progranulin, and C9orf72 genes that cause frontotemporal dementia. But these are all familial disorders, and so when it comes to rare genetic variation, this is kind of easier to study them because mutations typically segregate in the families, and so we can leverage that extra information. But when it comes to sporadic diseases like late onset Alzheimer's, studying rare variants becomes more challenging, basically because of two reasons. First of all, the low frequency, and second, because the cohorts that we usually have access to have low to moderate sample size. And these two factors typically translate into low statistical power to detect associations. Nonetheless, there are existing methods out there that can deal with this, but these association methods are mostly gene-based and univariate in nature. And most importantly, these methods lack the integration of information from other domains of biology, like, for example, molecular interaction, that can typically help to provide more meaningful results. And this latter point is particularly important in light of a recently proposed model. It's called the omnigenic model for complex traits and diseases, that basically postulates that most genes affect disease risk through highly connected cellular networks, meaning that potentially every gene in the genome, if affected by a mutation, can lead to a disease phenotype by virtue of their interaction landscape within the cell. And so one way we can implement the omnigenic model and see if it works is to use the so-called network propagation approach. This is an iterative procedure that mimics a wide range of real-life processes, like, for example, the spread of an epidemic in a community. And in our context, let's assume we have a gene interaction network, like the one depicted in the figure. And let's assume we have a single gene affected by a mutation pinpointed in red. So basically what network propagation does is to spread the effect of this rare mutation along the network edges, so that in this way, yes, so that after a few iterations, basically all the genes in the network have been affected by this mutation by virtue of the small world property of biological networks. So in this context, what network propagation allows us to do is to do two things. First of all, we can literally follow the biological pathways through which structural variation leads to phenotypes. And second, we can highlight genes that are vulnerable to the presence of mutations, not only in the gene itself, but also in the pathway. And so this leads me to the main aim of this study, which was to develop a gene-based association testing framework guided by this additional knowledge of tissue-specific interaction networks. And so we developed this pipeline that we termed NetPage, that stands for Network Propagation-Based Assessment of Genetic Events, that basically starts off with a subject times genes matrix of rare variant burdens, runs it through the network propagation step, and then uses the resulting smooth scores to fit an elastic net regression model with stability selection analysis in order to identify genes that are robustly associated with some imaging outcome. After extensive simulation, we have applied this method to data from the Alzheimer's Disease Neuroimaging Initiative. Specifically, we used genotype data from 708 subjects in the form of whole genome sequencing, and we focused on rare, exonic, non-synonymous, and deleterious variants that were mapped to 13,000 genes. As a genetic interaction network, we leveraged the hippocampus functional network derived by Casey Green and colleagues at UPenn. And as phenotype, we used whole hippocampus and 12 subfields segmented from T1-weighted MR images with free surfer and normalized by total intracranial volume. As for the results, here we can see the main result in the form of a modified Manhattan plot, where the y-axis represents the probability of a gene being selected by the sparse regression. And we can clearly see that there are 4 genes that clearly stand out in their association with the volume of the CA3 subfield of the hippocampus. Then after, on a closer look, we realized that 2 of these genes in the top row of the scatter plots, for those genes, the stability selection was clearly driven by a few outliers, so we chose to discard them and to focus on the other 2 genes, namely the CLU gene, clustering, and the OTUD4. And this is already an interesting result because we know that the clustering gene has already been implicated in Alzheimer's disease by means of common variant GWAS. So it's kind of interesting because it means that our method is actually picking up some true signal. Then, on a closer look, if we consider the gene burdens before and after the network propagation step, we can clearly see that the network propagation step enhances and makes more statistically significant the correlations between gene burdens and the volume of the CA3 field, which is also a proof of the added value of the method we're implementing. Then we performed some functional follow-up for the OTUD4 gene because that hasn't been implicated in Alzheimer's disease yet. And so we first turned to the Mayo Clinic temporal cortex gene expression dataset, and we found that the expression of OTUD4 is upregulated in the brains of patients with Alzheimer's disease, specifically in the temporal cortex, which is the closest region we could find to the CA3 field of the hippocampus. Also, we performed gene set enrichment analysis for the clustering gene, OTUD4, and their first neighbors in the interaction network that you can see depicted in the figure. And we found significant enrichments, significantly strong enrichments for gene ontology terms and gene sets closely related to Alzheimer's disease pathophysiology. And lastly, we turned to the literature, and we learned that this OTUD4 gene is involved in a biological process called ubiquitination, which is also disrupted in not only in Alzheimer's disease, but in a wide range of neurodegenerative disorders. And in particular, there's a rare mutation in this gene that has been shown to be causal for a rare form of dementia, also involving the hippocampus. So to recap a little bit, I would like to highlight the main contribution of this work, which are that, first of all, we performed the first computational proof of concept of the omnigenic model for complex traits and diseases. Also, we developed a multivariate association testing framework that integrates the so-called omics data in the form of molecular interactions. Also, we independently validate a known GWAS locus for Alzheimer's, and we also identified a novel locus as a hub of a network where the effect of rare variants accumulate. There are also some limitations for this work, namely from the computational side. For example, the benchmarking is not trivial, and we're also in need of a more thorough investigation of the parameter space for this method. With this, I would like to thank all our collaborators at UCL Center for Medical Image Computing and the Dementia Research Center, and also our collaborators at Stanford University, with whom we are trying to expand and validate this work in other cohorts. And if you have more questions, you can come find me at poster 1092 this afternoon. And I'd like to thank you for your attention, and I'm happy to take any questions.